So now I am going to discuss with you about the second part of this video lecture about the changes in the chromosome number, and I am going to discuss with you all about the polyploidy. In the previous video I have done about the euploidy concept, and especially about the monoploidy. Okay, now let us see about the polyploidy in detail. So, in polyploidy, organisms with more than two sets of chromosomes are known as polyploids. So, if the organisms have more than two sets of chromosome, then they are called as polyploid. Like for example, individuals having three sets of chromosome, that is two set diploid plus another set n, are called as triploids. Individual having four sets of chromosome, that is extra 2n, that means extra two sets of chromosome present in cell, are called as tetraploids. Now, depending upon the what is the source of additional chromosome sets, from where these additional chromosome sets comes from, depending on that, polyploids are of two types. One is called autopolyploids and another one is called allopolyploids. So depending on what is the source of that additional chromosome sets, polyploids are of two types. One is autopolyploids, another one is allopolyploids. Let us see in detail. What are autopolyploids? Autopolyploids composed of multiple sets of chromosome that has been originated from within the same species. So from that same species, your multiple sets of chromosome are being observed. Now this can occur by artificially induction of some chemicals like colchicine, or it can be done through internal factors as well. Whereas allopolyploids, if you see, allopolyploids are composed of chromosome sets from two or more different species. Like for example, when we are going to create a hybrid using the protoplast from the different plants cells and fusion them, fuse that protoplast with the help of certain process called protoplast hybridization, then we can make this type of allopolyploids. So allopolyploids composed of chromosome sets from two or more different species. Allopolyploids are also known as amphidiploid tetraploidy. It is also known as amphidiploid tetraploidy. So now I am going to discuss with you about the autopolyploids first. So I have said that autopolyploids are composed of multiple sets of chromosome that originate from within the same species, and how they are being created. So it is saying that these autopolyploid arise in any one of the following way. First way, by the union of the diploid gametes produced in the absence of meiosis or due to abnormal meiosis. So the first point they are marking that by the union of diploid gametes. How you can create diploid gametes? Obviously by artificial way, by using chemicals like colchicine to make chromosome doubling. So union of the diploid gametes, that diploid gametes produced in the absence of meiosis or due to abnormal meiosis. So one diploid gametes are having haploid sets of chromosome. These diploid gametes are induced to form 2n, and these diploid gametes are fused to form a tetraploid. This is one way that we can do. So union of the diploid gametes produced in the absence of meiosis or due to abnormal meiosis can lead to the formation of 4n, which is a tetraploid or you can say autotetraploid. Okay, this is the first way that we can do the autopolyploid. Second way is somatic doubling of the chromosomes. By somatic doubling of the chromosomes in a diploid zygote due to abnormal mitosis. So by doing abnormal mitosis in where? In the diploid zygote. Zygote are always diploid. And because the haploid cells, the gamete cells are fused to form that zygote. If that diploid zygotes are allowed to fuse, allowed to not fuse, allowed to undergo abnormal mitosis, then we can able to produce autotetraploid again. Okay, so the zygotes are forced to produce, so it has been cut off some place. I'm writing. So what happened? The zygote having two sets of chromosome. Now this zygote is forcefully carried out abnormal mitosis. So abnormal mitosis is carried out. Abnormal mitosis is carried out, and how they are carried out? By using colchicine. You can use colchicine, which disrupt the spindle fiber, and cause chromosome doubling. That leads to doubling of the chromosome. That cause doubling of the chromosome, and that leads to the formation of 4n, that is autotetraploid. So in this way it can be done. So by somatic doubling of the chromosomes in a diploid zygote due to abnormal mitosis, that leads to the formation of autotetraploid. Third way is by union of the haploid gametes with the diploid gamete. So union of the haploid gametes are always haploid, and we are uniting that haploid gametes with diploid gametes. The diploid gametes are carried out by abnormal meiosis. They are fused to form triploid, which is called autotriploid, right? Fourth way is by a cross between a tetraploid with a diploid parent. So a tetraploid plant is crossed with a diploid parent. So a tetraploid, as it is given in the figure, I have tried to show you. So this is a tetraploid parent, this is a diploid parent, this is their gametes. So obviously a tetraploid parent will have diploid gamete, whereas a diploid parent will have a haploid gamete. They are allowed to fuse to form triploid, which is the autotriploid. Autotriploid. Okay. So in these 3-4 ways we can go for autopolyploidy. Okay, where multiple sets of chromosome originating from within the same species. Now it is saying that autopolyploids are relatively rare in nature. These can be produced by artificially. They are very rare. You cannot find them naturally, but it has to be induced them. Like for example, the common bread wheat that we eat, okay, that is a hexaploid. It has been created by fusion with many wild species of wheat, Encorn wheat group, Emner wheat group. You will see them in this video. So autopolyploids are relatively very rare in nature. They can be produced by artificially, like natural autopolyploids are the doob grass. Now in Bengali, doob grass is all known as durba ghas, that we know. Now this doob grass is actually a example of autopolyploid. Scientific name of doob grass is Cynodon dactylon. It is a autotriploid, that means it has 3 sets of chromosome. Rye, you know we eat rye. Sickle cereal. Rye is an autotetraploid, that means it has 4 sets of chromosome. Okay, rye is actually a member of wheat tribe, which is closely related to barley and wheat. Okay, so naturally we can find these auto polyploids very rarely, like Cynodon dactylon, which is a autotriploid, then the sickle cereal, which is also a autotetraploid. I hope you have understood up to this much. Autopolyploids are induced by treating the zygote with colchicine. So nowadays we can induce to form autopolyploids by treating the zygote with colchicine, such as banana, in treating the zygote with colchicine in banana plant, in datura plant, potato plant, and coffee, and many other crops as well. Now it is saying one important thing, that by using colchicine we can treat to form these autopolyploids, because colchicine what they do, I have already said that what colchicine do is, if this is the cell, during cell division what happen, the mitotic spindle, which is made up of microtubules, they are needed for separation of the chromosome towards opposite poles. So the chromosomes, they align at the center, known as the metaphase plate. These chromosomes has to migrate to the opposite poles, so that they can equally divided among the daughter cells during mitosis. Now what does colchicine do is, colchicine disrupt this mitotic spindle. It disrupt this mitotic spindle so that the chromosomes cannot able to migrate towards the opposite poles, and thus the chromosome remain in this phase. It remains in this phase within this cell, and thus chromosome doubling occurs. In this way we can induce autopolyploidy in the same cell. So by treating the zygote with colchicine in banana plant, datura plant, potato plant, coffee plant, we can make autopolyploids as well. Other chemicals are also there. We will name them later on. Now it is saying that polyploids with odd number of chromosome sets are sterile or highly infertile. Why? Why polyploids having odd number of chromosome sets, that means triploid, odd number of chromosome, pentaploid, heptaploid. So polyploids having odd number of chromosome sets are sterile in nature, or they are highly infertile, because they are gametes which will be produced from these parent cells. These gametes will be aneuploid in nature, means they will have missing of a chromosome segment or a whole chromosome, or they will have an additional chromosome. Why this occurs? So polyploids having odd number of chromosome sets are sterile, or they are highly infertile. Why? Because the gametes and offsprings are going to be aneuploid. The gametes will receive either an extra chromosome, or it may lose a chromosome segment. Why this occurs? Because paired homologues segregate to the opposite poles, but unpaired homologues, they have to pass through the either pole randomly. So if I can draw a diagram here, you know the chromosome look like almostly like this. Chromosome looks like this. It has a centromere, and these are the arms of the chromosome. P arm is the small arm, whereas the Q arm is the large arm. Now this chromosome during S phase of the cell cycle, the DNA duplicates itself, as you have learned in your molecular biology that DNA replicates. Actually the chromosome is making its duplication so that during mitosis it can equally distributed to the opposite daughter cells. Now these chromosome, they get doubled to form bivalent. They form bivalent like structure, which looks like this, right? In the center there is a centromere. I am doing with different color. So this is a bivalent. This is a bivalent. Now when these bivalent are going to be separated by the mitotic spindle, this time attachment occurs, mitotic spindle, they can easily migrate to the opposite poles. They can easily migrate to the opposite poles. But what about trivalent? What about univalent? Suppose one extra chromosome is remaining. Suppose one extra chromosome is there of that same homologue. Like for example, suppose this is the chromosome number 1. This is the chromosome number 1 and this is also the chromosome number 1. So 3 sets of chromosome 1. What about this? Where it will migrate? It has to migrate either on this way or on this way. So the paired homologues, they will segregate, they will separate equally to the opposite poles. But what about the unpaired one? The unpaired homologues, which are the univalent, they has to pass to the either pole randomly, and thus what happen, those polyploids which has odd number of chromosome sets, their gametes going to be receive either an extra chromosome or loss chromosome. Due to the unpaired homologues, they cannot, they migrate randomly to the opposite poles, to the either poles, and the result of the is the meiotic products with chromosome numbers intermediate between the haploid and diploid number. Such genomes are termed as aneuploid, because in aneuploid you will get either extra set of chromosome or you will lose a chromosome set. Okay, that's why it is saying that polyploids having odd number of chromosome sets, polyploids having odd number of chromosome sets like triploid, pentaploid, heptaploid, these odd polyploids, their gametes are going to be sterile, because their gametes are having the aneuploid like conditions. One of the gametes will have an extra chromosome, another gamete will have loss of that chromosome. This type of condition will be created. Okay, I hope you have understood this part. Let us move to the next part. It is saying that aneuploid gametes do not give rise to viable offsprings. Okay, because of that having that extra chromosome or loss of that chromosome part. Autotetraploid arise by doubling of a 2n complement to 4n, and this can occur spontaneously or by artificially. So the autotetraploid, it can arise by doubling of a 2n complement. So the 2n, which is a diploid condition, if it is induced to form 4n by externally or by spontaneously internal factor, this can be done by artificially or by spontaneously. Now in case of 4n, I have said about the odd number of chromosome sets, that their gametes are going to be sterile. But what about the tetraploid and octaploid? We don't have seen octaploid, we have seen the hexaploid. Up to hexaploid we have seen. So as 4 is an even number, autotetraploids can have regular meiosis, because by the way we get two pair homologs here. 4n means what? 4n means what? Each of the chromosomes will have 4 in number, and 4 in number means two pairs of each chromosome. That two pairs will be chance, there is a chance that the two pairs is migrating, segregating to the opposite poles in equal number. So as 4 is an even number, autotetraploids can have a regular meiosis. The crucial factor is that how these 4 chromosomes of each set, how that 4 chromosomes pair and how they are segregating. This is the main thing that we have to know. Okay, so those polyploids which have 4 sets of chromosome or 6 sets of chromosome, they have a regular meiosis and they can have viable gametes that will further fertilize to form a new being. But the main problem is how these chromosomes, if it is a tetraploid, how these 4 chromosomes of each set pair and how they are segregating, how they are distributing to the opposite poles. Now during meiosis, the 4 homologous chromosomes may pair as 2 bivalent, just I have shown you the figure, or as quadrivalent, means all the 4 chromosomes migrate to one pole to form a tetraploid, and these can yield functional gametes. So two things can occur. One thing is that good positive thing, that the 4 homologous chromosomes, they may pair as 2 bivalent. 2 bivalent can be formed like this. One bivalent, another bivalent. So 2 bivalent migrate to the opposite poles equally, or as a quadrivalent, all the 4 chromosomes are moving in one pole to form tetraploid set, and that can yield functional gametes. Let us see the figure now so that you can able to understand. I am not showing this figure, I will show the book figure. Okay, so you can see the 4 chromosomes here, 1 2 3 4, and the 4 chromosomes are aligned in this way, you can see, and that 4 chromosome of that set is moving to the opposite poles to yield functional gamete, or they can move the whole chromosome, the whole 4 chromosomes are moving to one pole, which is called as quadrivalent. But however, what happened, the 4 chromosomes may also pair in a univalent trivalent fashion. That's the thing. So in a univalent trivalent fashion, the 4 chromosomes may move. Univalent trivalent fashion means in the 3 chromosomes of that same chromosome set, is moving to one pole while only one chromosome is moving to the opposite pole, and that univalent trivalent combination yields non-functional gametes. So if you see in this figure, you can see, don't look the left figure now, look at the right figure. All the three chromosomes are moving to one pole, whereas one chromosome is moving to the opposite poles. That is a univalent trivalent combination. Here 4 chromosomes should be present, it is not shown in the diagram. So this may occur, and the same thing occur in triploid actually. Those who are having odd sets of chromosome, that's why here triploid has been shown. If you see the triploid, that means having 3 sets of chromosome. So 3 sets of chromosome means each chromosome have 3 in number, 3 copies of each chromosome, that's why 3n, 3 sets. So how will they pair? 2 chromosomes are moving to one pole, whereas the another chromosome is moving to the opposite pole. So obviously aneuploid like conditions may occur. Similarly in tetraploid, those who are having 4 sets of chromosomes, they can also undergo this type of conditions where 3 chromosomes are migrating to one pole and 1 chromosome is moving to the opposite pole. That univalent trivalent combination can also give to non-functional gametes. So those who are autotetraploid or autohexaploid, they are semi-sterile, but not that of autotriploids. It is much less sterile than autotriploids. So just go through this diagram. Here you can see meiotic pairing possibilities in tetraploids. Each chromosome is really 2 chromatids. The each chromosome that has been shown is made up of 2 chromatids. For simplicity, it has shown in this way. And the first way they show that in a case of 2 bivalent, they are moving to the opposite poles very well, or whole as a quadrivalent, they are moving to one pole. Means either pole, either in this pole or in this pole, all are moving. In both these cases, the gametes are going to be viable. Gametes are going to be viable. But if they move in a univalent trivalent fashion, that means 3 chromosomes in one pole and 1 chromosome in the opposite pole. See, this chromosome is moving to this pole and the rest of the 3 chromosomes is moving to this pole. The arrow mark has not been shown. Then it leads non-viable or non-functional gametes. Same case we have seen in case of triploid, those who are having odd sets of chromosome, triploid, pentaploid, heptaploid. This type of polyploids, having odd number of sets of chromosome, they also undergo this type of sterile condition, aneuploid like conditions. Okay, I hope you have understood this part. Now comes the allopolyploid. Allopolyploid are composed of chromosome sets from 2 or more different species. So allopolyploid, they are being hybridized actually by from the 2 or more different species, we combine them to form allopolyploid. For example, allotetraploid between 2 species A and B will be formed if F1 hybrid AB of these 2 species undergo chromosome doubling and produce diploid gametes. So just like 2 species, species A and species B, they fuse and they undergo chromosome doubling to form allopolyploid. In some cases, diploid cell of the hybrid on set of the mitotic division undergo chromosome doubling but do not divide. Such cell is also called tetraploid. So in some cases, the diploid cell of a hybrid, suppose we have created hybrid by fusion of the two protoplasts of different species. Now if that hybrid cell during the onset of mitotic division, if it undergoes chromosome doubling but without cell division, if it undergoes chromosome doubling, such type of conditions will be called as allotetraploid as given here, or you can say allopolyploid like conditions, because we have fused them to form a hybrid cell. They were You have fused them to form a hybrid cell, and if this hybrid cell, now this is taken from different species, and that hybrid cell undergo chromosome doubling to form 4n, that will be called as allotetraploid or allopolyploidy. Now it is saying that treating a sterile hybrid with colchicine, treating a sterile hybrid with colchicine greatly increases the chance of chromosome doubling sets. Why? Because in this treatment the S phase of the cell cycle occurs, but not the chromosome segregation will occur. As the cell enters the telophase, a nuclear membrane forms and the entire double sets of chromosome that leads to the tetraploid like condition with exactly 4 copies of each type of chromosome. So this type of sterile hybrid can be created, like for instance the Raphanobrassica. It is the first known allopotetraploid which has been prepared by the Russian geneticist G. Karpenshenko. Now this Raphanobrassica is a hybrid of Raphanus sativus, which is radish that we know, and Brassica oleracea, which is the common name of cabbage. Two are different species, two are different genera. They are distantly linked, very distantly linked. So when they are fused, they have the same sets of chromosome actually. That's why they have used these two plant species for allopolyploidy, and they have prepared the diploid gametes of the F1 hybrid. Let us see how they have prepared the F1 hybrid. Both radish and cabbage are diploid, and each radish and cabbage have 18 chromosomes. That means their 2n is 18. That means their haploid number will be 9. So their gametes will have 9 chromosomes. Union of these gametes, fusion. Now it can be fused only by certain other process because they are distinctly linked organisms. They are distinctly linked species. So union of these gametes produce the F1 hybrid having 18 chromosomes. These hybrids are sterile because the 9 chromosomes of radish will not able to pair with the 9 chromosomes of cabbage. So both radish and cabbage have 18 chromosomes in their somatic cell. So their gametes will obviously contain 9 chromosomes. We try to fuse, we try to unite these gametes to produce F1 hybrid having 18 chromosomes. Now these hybrids are sterile because the 9 chromosomes of radish will not able to pair with the 9 chromosomes of cabbage. So what does we do? Sometimes what will happen, viable pollen and ovules having 18 chromosomes, that is diploid gametes, can be achieved from spontaneous accidental chromosome doubling of the F1 hybrid. So it is saying that sometimes viable pollen and ovules having 18 chromosomes, which is called as diploid gametes. So how the pollens and ovules have 18 chromosomes, the n, which was n, it becomes 2n. How it is possible? By chromosome doubling, and that can be done by colchicine treatment that we know, or by spontaneous doubling. So sometimes pollen and ovules having 18 chromosomes, that is diploid gametes, are formed from spontaneous accidental chromosome doubling of the F1 hybrid. Spontaneous accidental chromosome doubling is carried out in F1 hybrid so that it becomes now 36, because if the gametes become 2n, this also becomes 2n, become, that means 18 18, and what does 18 into 2 means? 36. Now we have 36 chromosome, we become allotetraploid. So these gametes are derived from spontaneous accidental chromosome doubling to form a sterile hybrid, presumably in the tissue that eventually become germinal and underwent meiosis as well. And this fusion of these diploid gametes produce allotetraploid. That means now it becomes 36 chromosome. In its diploid cell, the gamete will produce 9 chromosomes with 18 chromosomes of radius and 18 chromosomes of cabbage. So in this way, your Raphanobrassica is being prepared. Now this is the flow chart that has been shown here. Raphanas species, full name is Raphanas sativus, having 18 number of chromosomes, and Brassica oleracea, which is cabbage, is it's also having 18 sets, 18 chromosomes. Their gametes will have 9 chromosomes. Obviously they are trying to fuse to form a F1 hybrid. Now this can be done artificially by somatic hybridization technique, or by protoplast fusion technique. Okay, and after formation of this hybrid, it is saying here that sometimes, now this F1 hybrid is sterile, but it is saying that sometimes viable pollen and ovules having 18 chromosomes are being formed from spontaneous accidental chromosome doubling of the F1 hybrid. So if the F1 hybrid are sterile in nature, it is saying that some pollens of from that F1 hybrid, they become viable and they are formed from spontaneous accidental chromosome doubling of the F1 hybrid. And spontaneous accidental chromosome doubling is carried out if in F1 hybrid, so that it becomes 36 chromosome now and can able to produce diploid gametes as well. Further we can go for another process, like we can treat them with colchicine. Here we can treat them with colchicine. We can treat with colchicine here. So it has already been written, colchicine treatment to produce their somatic chromosome number as 36. So their gametes will be 2n 18, and thus we prepare a new variety of species called Raphanobrassica, which is a tetraploid hybrid. But unfortunately, what does Karpenshenko wanted was, he wanted to make such plant species which will have the leaves of cabbage, and the root side there will be a radish. Okay, but unfortunately for Karpenshenko, his allotetraploid level has the roots of a cabbage and the stem of a radish. So this is that thing. I think you have understood. Now, what is the significance of polyploidy? It not only increase the size and vigor of the plant. Polyploidy not only increase the size and vigor of the plant, but it also changes the size of the fruits and pollens. You must remember, more number of sets of chromosome, more number of sets of chromosome means there will be more increase in the height, more increase in the weight, more increase in the size of the fruits and pollens. That's why hybrid plants are being prepared. So there is a correlation between the number of copies of a chromosome set and the size of the organism and its component parts. Typically, a tetraploid organism looks very similar to the diploid, but tetraploid are much bigger in its component parts. So, the higher the polyploid level, larger will be the size of the plant. So, size is proportional, directly proportional to the level of ploidy, how much set of chromosome is present. Second importance of this polyploidy concept is that with the help of polyploids we can change the season blooming and fruiting and change in the annual plants into perennial plants. Annual plants are those plants which grows once in a year and they will die, like for example cabbage, your carrot, your cauliflower. But nowadays you can see that these plants are being used to form perennial plants which can grow for up to 2 years, 3 years, so on. This can be achieved through polyploids. Okay. New varieties of plants for improvement of the crops in terms of resistance to disease, high yield, better quality can be obtained by interbreeding and polyploidy. So, new varieties of plants for improvement of crops in terms of resistance to disease, higher yield, better quality can be obtained by interbreeding and polyploidy. How new plants can be achieved? One variety has been shown here, Triticum aestivum, the common bread wheat that we eat. It is an example of a new variety. Now Triticum aestivum is 6N, that means it is a hexaploid. It has 42 chromosomes and it is a allopolyploid, that means it has been prepared from different species of wild wheat group. Okay, how it is possible? Now let us see about the two wheat groups that we want to discuss. First is N-corn wheat group. N-corn wheat group is represented by two species, Triticum monococcum and Triticum agilopoids. Triticum agilopoids and Triticum monococcum. Now these are the wild type diploid wheat. Wild type means primitive or dominant. It has 14 chromosomes in its diploid condition. The problem is that this wheat cannot be suitable for human consumption because the grain is very tightly packed in glumes. Glumes are the modified leaf or scale which is small with the flower and it cover up your seed part. It cover up your seed part. So the glumes are so tightly packed the grain that it is very difficultly we can open out that carry open that we can isolate that grain from the glumes. So they are very problematic. Secondly, they are not so much of nutritious. So these N-corn wheat group are tried to cross pollinate with distinct variety of goat grass known as Aegilops peltoides. You can see in the next page I have seen. So this Einkorn weed group, which is Triticum aegilopoides, or you can use Triticum monococcum, it is trying to cross pollinate with a distinct type of goat grass known as Aegilops peltoides. It also have 14 chromosomes, and they are fused to form a hybrid. That hybrid is sterile, obviously, due to genome incompatibility, chromosome incompatibility, where we force to induce chromosome doubling. Spontaneous chromosome doubling can be carried out by using colchicine, and we prepare a new variety of wheat, Triticum dioicoides. Okay, this Triticum dioicoides is actually an Emner wheat group. Now it falls a Emner wheat group. So you will see the name here. Emner wheat group includes 7 allotetraploid species with chromosome complement 2n equals to 28. And the most two important species of tetraploid wheat are Triticum dicoccum and Triticum durum. Emner wheat group has evolved by hybridization of the Einkorn wheat group and wild species Aegilops peltoides, which is a goat grass. So the Einkorn wheat group variety is crossed with this goat grass Aegilops peltoides to form this hybrid, and this hybrid is forced to induce chromosome doubling by colchicine treatment to form this type of Emner wheat groups called as Triticum dicoccoids. Dicoccoids. In the next stage, or dicoccum. In the next stage, we get a vulgar wheat group. In vulgar wheat group, simply we cross this Emner wheat group. From the previous page, we get Triticum dicoccum, which is a Emner wheat group. It is again fused or cross pollinate with a type of goat grass called Aegilops squarosa. So Triticum dicoccum, which is an Emner wheat group, is crossed with Aegilops squarosa, which is a common goat grass, to get an allohexaploid, which is your Triticum aestivum, which is the common bread wheat that we eat, that we consume them as flour. Okay. So in this way we can get new varieties of plant for improvement of crops in terms to resistance against disease, higher yield, better quality can be obtained by interbreeding and polyploidy. Okay. Now I have said about colchicine, but other than colchicine, we can also use oryzalene, we can use trifluoralin for induction of chromosome doubling. All these chemicals, they inhibit the formation of spindle fibers during mitosis, but not doubling of the chromosome. Okay. Now it has also been said that we can also change the chromosome number by extreme temperature, like maize and other plants respond to changes in temperature. Hence, when they keep on high or low temperature, it inhibits the spindle formation, thus tetraploid cells can be formed. So this is about your polyploidy. In the next video I will try to discuss with you about the euploidy part, where you will see in euploidy you will see the trisomy, the tetrasomy. Okay. And related with the disease, actually I will mainly discuss the monosomy. And also one important thing that is given in your syllabus, the non-disjunction part. What is non-disjunction? Simply it means if the chromosome not able to segregate properly to the opposite poles, that is your non-disjunction. Okay, and bivalent can cause can overcome this non-disjunction part, but those who are polyploids, they cannot undergo through this disjunction process, they undergo non-disjunction part. So that's all for it for today's lecture.